If you are very unwell in hospital, 2 days is too long to wait to know if you're on the appropriate antibiotics. So now we had a really great way of removing human DNA from clinical samples, and it was efficient and fast. So speed is key to everything that we were trying to do with metagenomics. We were trying to get a test, a complicated new technology, to work quickly and to provide the data that we needed in a time frame that was faster than culture. We needed to do all this within a day. We needed to provide same-day results to the clinician, and we needed to do that with a sequencing technology. So this hadn't been done before. Now, blood wasn't an easy sample to work in, and it was a very challenging ratio, pathogen to human. So we moved on. We moved into urine, and then we moved into respiratory tract infection. These are easier sample types. Urine was easy and quite nice to work in, but the economics don't really work out for using metagenomics in urinary tract infections. So you need, a clinical syndrome or disease like pneumonia, where it causes a lot of deaths in hospitals, and healthcare systems are willing to pay more money for a test in that particular type of infection. So we started working on respiratory tract infections. There are more bacteria in the lung, and when a patient is infected, there are more. There's around the same number of human cells, so that ratio isn't as bad. And when you remove that human DNA, you end up with more nucleic acid to work with, and it's just an easier sample type for metagenomics. So that application started to really take off. We started working on that as part of a program called the INHALE trial, where we trialed the BioFire film array for pneumonia, the Curatus Univero test, and metagenomics. And we tested all three of those technologies, and we chose the one that was most implementable and best at the time, which was the BioFire film array. But metagenomics did really well. The problem with metagenomics is it wasn't implementable, right? So it was too challenging to do in the lab. And I think that's true to this day. So I'll talk about that a little bit later. But basically, now we had an indication or an application that looked like it was going to work, and that was respiratory. And so we needed to get that turnaround time down into a day. So we needed to look at each part of the process and optimize each part of the process. So the homogenization of the sample to begin with, moving into the host depletion step, then moving into DNA extraction. So we wanted to keep those times down as much as possible. We've actually brought the time down for host depletion down to 15 minutes. We do it in one tube, and it's really quick and simple, and we still use the same enzymes. We also started using a library preparation kit from Oxford Nanopore, which was really fast, which was the rapid PCR barcoding kit. And then we were able to add a number of samples, so multiplex samples on the MinION flow cell to keep costs down. And we were able to reduce the time we needed for sequencing because the Oxford Nanopore flow cells got better and better over time. And then eventually we came out with a protocol that was able to work within about 6.5, 7 hours. And that is where we needed to be. So we could get a sample in the morning, and we could provide a result to the clinician by the end of the day. We could give them the pathogens that were present in the sample and some information on antimicrobial resistance. We could follow that up later with some additional AMR data as we got more data off the sequencer, but we certainly had some good information to provide on the same day. So we finished the INHALE trial, and we published a paper from that work, from the metagenomics test that we developed during that time. And one of my PhD students, chemist Cheryl Lampus, she was the lead author of that. And we published that paper in Nature Biotechnology. And that really made a bit of a splash in the field. People were very interested. Just a year or two later, Themis finished her PhD, and she was hired by Jonathan Edgeworth at GSTT. And that was just 2 weeks before the start of the pandemic. I went down to help out at GSTT with some diagnostic development work when the pandemic started. And so we talked to Jonathan, and we decided that it would be a good idea to try to implement metagenomics for respiratory infections of COVID-19 patients while they were in the ICU in St. Thomas's. And we wanted to look for co-infections with bacteria or fungi, and there was a theory at the time that there might be a co-infection etiology to some severe presentations of COVID. That turned out not to be the case, but the metagenomics testing was very useful anyway, and we published a nice paper on that. Not long after that, I decided to make a move out of academia and into Oxford Nanopore. I'd worked with them for about 8 or 9 years at that point. I was very interested in the technology and I really wanted to drive it forward. So I brought some of my techniques with me into Nanopore and developed them further. So I developed my respiratory metagenomics test further within Nanopore. We added the ability to detect viruses, and we made it as robust and as fast as we could. And we released a protocol to the community about a year ago now, and that protocol is being widely used in the community. So I guess that brings me up to my recent move in June to the Ellison Institute of Technology in Oxford. And so what we're doing at Ellison is that we're going to take metagenomics as an approach and we are going to automate it. And I think that's really the missing step. I mentioned earlier that about implementation and why metagenomics is difficult at the moment to implement. And that's really because it's a very long procedure, which is quite laborious. And so maybe there's 3 or 4 hours worth of lab work with people in white coats trying to do somewhat complex molecular biology. We need to take that and simplify it and fix that problem and move metagenomics in the same direction as qPCR did, into cartridges, sample to answer. And so that is the challenge that we're going to address at Ellison Institute of Technology. And with my colleague, James Clark, we're going to take metagenomics, the approaches that we've developed, improve them, we're going to automate them, we're going to put them into cartridges in machines that will go from sample to answer. In the first instance, we will certainly go from sample to library, and then we will transfer the library onto a MinION flow cell, run the MinION flow cell, analyze the data, provide the answer. And so that work is what we plan to do over the next few years, and I think that's key if we're going to get metagenomics implemented in hospitals across the world widely, just like we have implemented qPCR widely using sample to answer devices.